This presentation looked at the impact of acetylcholine transporters on heart rate variability. There were 2 papers analyzed for this. One looked specifically at a single nucleotide polymorphism, while the other looked at 8 different genes and included a variety of variation. There was a 10-year time gap between the studies, so there was some variation in technique and in the types of measurements used to analyze heart rate variability. So heart rate variability is a metric similar to resting heart rate or heart rate, however it measures the time in between the beats. So for example, if your average heart rate was 60 beats per minute, there wouldn't necessarily be 1 second in between each of those beats. It could be 0.9 seconds for some or 1.1 seconds for other beats. And this is largely related to the cardiac autonomic regulation, or the body's readiness and response to stress. A higher variability indicates that the body is ready to take on stress, while a lower variability would show that the body is still working to recover, essentially. A low HRV has been associated with a poor health diagnosis across a variety of studies. Some of the common metrics for analyzing heart rate variability include looking at the high frequency domain, the low frequency domain, and the ratio of these 2 areas. These are more frequency domain based, and then the alternative is the root mean square of successive differences, which is more of a time-based measurement. Acetylcholine is a major transporter in the sinoatrial node in the heart. The acetylcholine transport is regulated in a rate-limiting step by the CHT1 gene, which is the gene analyzed in both of these studies. One of the metrics used to evaluate HRV is RSA, which accounts for respiration, and this is also factored into both of the studies. So in the 2005 study, there were 413 participants who were measured using an electrocardiogram for 5 minutes. And then they were genotyped for the single nucleotide polymorphism that was in question, which was located in the 3' untranslated region of the gene. In the 2014 study, over 400 single nucleotide polymorphisms were analyzed across more than 8 different genes, including the one that was analyzed in the 2005 study. And the patient pool for this study was over 6,000 in total and was conducted with a stage 1 group of 3,429 individuals and a second verification stage of 3,311 individuals. The methods for the HRV measurement were slightly different for this second study, as it was composed of different subgroups for both the initial stage and the secondary stage, but the technology used in all of these groups was equivalent or better than the one conducted in 2005. So the findings for the 2005 study indicated that of the 413 individuals, 76% of them had the G phenotype, or the more common of the 2 phenotypes for that position, while 24% had the T allele, the least common of the 2. And so what they determined from this is individuals who had the T allele were found to have a greater parasympathetic activity and a lower sympathovagal balance. Meanwhile, in the 2014 study, of the 447 SNPs that were analyzed, only 25 were found to have a p-value less than 0.05 or were found to be significant. And even of these 25, there were not any that passed the second stage of this study, as there was not found to be an association. And in the meta-analysis, there was still not enough evidence and data to prove an association for any of these SNPs. So for further research, the SNP that was analyzed in both studies was looked at through the GTEx portal to see if there were any associations for this SNP, and there were no SQTLs or EQTLs associated with it. When looking at the genomic region surrounding the SNP, there were quite a few SNPs that were also looked at in the multi-gene study. And as you can see here on table 9, there are multiple SNPs that are in the tail end of the SLC5A7 gene, which was the gene in question that controls the acetylcholine rate-limiting step. So of the comparative analysis, the biggest difference between the two studies were the sample size and also the metrics used for the studies, as the first study used more frequency domain metrics, while the second study used more time-based measurements. And despite the 2014 study concluding there were not significant associations, there are a lot of SNPs that were looked at, and of the top ones analyzed out of the 440 genes, a lot of them were from the 3' untranslated region of the CHT1 gene, which does spark interest for where the underlying genetic cause is for variations in heart rate variability. Heart rate variability is a metric that's largely associated with heritability, so there is a large genetic factor to this, but despite that, it is such a complex trait that it can't be pinned to certain areas as easily as other traits within the genome. And heart rate variability is, in a broader significance, is an important metric, as it can be an indicator of health at a broad level. And the prevalence and importance of HRV is only increasing with the use of wearable devices that are now being able to transmit a reliable HRV signal. The more we can learn about HRV and its implications to various diseases and how it's varying between individuals and within an individual, the more this information will help us in order to prevent and treat chronic diseases that may be associated to HRV.